The last piece of the puzzle to talk about when we're talking about moving things into and out of the cell is talking about really big things. We talked about very small things that have no charge when we talked about diffusion across the membrane. We talked about using transporters to move other substances, ions like sodium, potassium, bigger molecules like glucose. But when we get to really big stuff, like here we can see a white blood cell engulfing bacteria, a whole other cell, then we have to have a different system of moving things into and out of the cell. So we're going to talk about these different processes. They have different names to them, but let's start with the basic. When we're moving big things, we have two directions they could go. We could bring things into the cell, and we could bring things out of the cell or send them out of the cell. We call these processes endocytosis, that's bringing into the cell by membrane budding. I'll show you what that means in a moment. So endo means within, and this is cytosis, is bringing inside the cell. Exocytosis is sending things out of the cell by membrane budding. Exo means outside. So this is sending things out of the cell. All right, let's look at these processes and be aware that they have slightly different names. So there's different types of endocytosis. One type of endocytosis is called phagocytosis. Phago means eating, so this is cellular eating. In this case, here's what I meant by membrane budding. So here's the plasma membrane of the cell, and here we have a large food particle or something that it wants to bring into the cell. It will actually cause the membrane to bulge inwards, forming this little invagination here, and then the particle is surrounded and it buds off as a little vacuole of membrane that brings it inside it. Now, there are many single-celled eukaryotes that eat bacteria as food in this way, but also a very important phagocytic cell in your body are white blood cells. These will engulf bacteria and then break them down and destroy them. So things like lysozymes break the bacteria down inside of the cell. So here's a phagocytic cell. It sends out its little bits of cell and engulfs it here and then brings it inside, and the lysosome fuses with this and it breaks down the bacteria in there. Okay, so that's phagocytosis. That's one type of endocytosis, usually for large objects that are other cells or things that might be food products or pathogens we want to destroy. There's also a process called receptor mediated endocytosis. Here, the process is mediated by a receptor. Okay, the name says it all, but what does that actually mean? We have this membrane here with a bunch of different receptor proteins on the surface. And these cell surface receptor proteins are looking for a specific signal. In this case, the star-shaped molecule. They will bind to the star-shaped molecule and cause this invagination, this inward folding, and bring in those star-shaped molecules into vesicles here, which will transport this substance into the cell. Now, this is a very efficient process of bringing things into the cell because it only brings in the thing you want, right? This specifically binds to the receptor here, and it only brings that in. All right, so this is a very efficient process, but of course, there are things out there in the world that will try to trick our cells, including viruses and bacteria that can exploit this receptor-mediated process. So you may have heard of this virus called SARS-CoV-2. It causes a disease called COVID-19. It has a spike protein, a little protein on the outside of its viral particle that binds to a receptor in our cells. Particularly this receptor, which is called ACE2, is found in lung tissue and lung cells. So this spike protein binds to the receptor, and the receptor is tricked by this and goes, that's exactly what I was looking for. Come on in, and it brings the viral particles in. Okay, ACE2 is normally looking for a different signal, but in this case, SARS-CoV-2 has evolved a shape that tricks the receptor. And here you can see an electron micrograph of these viral particles being brought into the cell by this receptor-mediated endocytosis. So We then hopefully evolve a different ACE2 so that this no longer is there, but then this will evolve a different shape and the arms race will continue, right? Or hopefully we can have something like vaccination where we build up antibodies against the virus and protect ourselves before it can attach to this. So this ACE2 receptor, luckily, is not highly expressed in young children. That's why during the COVID-19 pandemic, we saw very few children dying from this disease. It tended to be older people with weaker immune systems. So we express this ACE2 receptor in our lung tissue, and that's why we got respiratory distress, basically, because this is a respiratory virus. But ACE2 is expressed in other places, including reproductive tissues. We saw links between COVID and decreased fertility rates and things like that. And brain tissue. We're still learning where this ACE2 receptor is expressed and what it does in these different tissues. So we have still a lot to learn, actually, about what could be the long-term effects of being infected with COVID on other parts of the body beyond the lungs, right? We saw the immediate effects of people basically dying from suffocating because of the lung infection, but we don't know what other things could be waiting down the line. All right, so those are two. There's one final type of endocytosis. I teach this because this is always taught, but I'm going to tell you that I don't have a really good example of where this occurs, but you might be asked this on some sort of standardized test in the future, like an MCAT or something like that, so I have to teach it. This one's called Pinocytosis, and this is cellular drinking. It brings extracellular fluid and the molecules in it into the cell. So here you can see there's a bunch of extracellular fluid and these molecules are in it. So by budding in here, the fluid is brought in with the molecules that were in the fluid. The molecules don't bind to a receptor, so it's not as efficient as that previous process of receptor-mediated endocytosis. Again, I have looked high and low. I remember learning this when I learned this stuff. I have always taught this in every course that I've taught this in, and it continues to be in textbooks and on standardized tests, but I don't have a good example. I looked high and low and I could not find one, so I apologize for that, but just know that pinocytosis brings liquid in. Okay, so if you can bring things in, you're probably going to want to move things out of the cell, right? So substances that are too big to move through transport proteins need a way of getting outside of the cells. Waste materials often are things that we want to get out. Secreted proteins like hormones and neurotransmitters, we need to move those outside of the cell, and other protein molecules. To do this, we have vesicles that have these proteins packaged up inside of them, or whatever they're moving out, and those vesicles go and fuse with the membrane, releasing their contents outside of the cell. It's basically the reverse of endocytosis. So these two processes, endocytosis bringing things in and exocytosis bringing things out, means that our membrane is actually constantly cycling between different parts. We have plasma membrane that's moving inside, it's going to lysosomes, it's coming back out through exocytosis, it's moving to the Golgi, then things are moving out through transport and secretory vesicles back to the plasma membrane, and this whole process is cycling the membrane around. That's the beauty of the phospholipids, right? There's like no rigid bonds holding them together, so they can break off and come back and fuse together in a very kind of fluid system here. Okay, that is our moving things into and out of the cell. Let's go ahead and do a review question here before we finish up. I have an example here of some red blood cells. In these red blood cells, water is being pulled into the cells, causing them to burst. What type of solution is surrounding these cells? Is it hypotonic, isotonic, or hypertonic? Go ahead and think about that. Pause the video. All right, so the solution outside has water that's being pulled into the cell. So that would mean this is in a hypotonic solution. the liquid outside has less solute dissolved, so the liquid is trying to move into the cell to lower the concentration of the solute that's in there. That pulls the water into the cell and causes the cell to burst. All right, so we've done it. We've talked about moving things into and out of the cell. We talked all about membranes, everything from diffusion and facilitated diffusion, active transport, up to bringing large things in, these large forms of active transport called endocytosis, right? That brings big things into there. Talked about phagocytosis, cellular eating, receptor mediated endocytosis, substances bind to surface receptors, that brings them in very efficiently, and then pinocytosis, cellular drinking, brings the liquid with molecules in. Of course, if we can bring it in, we've got to send things out as well. Exocytosis, send things out of the cell by vesicles fusing with the plasma membrane. All right, that is it for Chapter 5 and our second unit. Nice job making it through.